And we've got two great speakers. I've got a little bit of introductory comments about patient registries. So the agenda that we're going to go through is we're going to talk about the learning objectives for tonight, the types and purposes of registries, and then our speakers are giving you very real-world examples from other conditions. And then we'll talk about the state of CPRN registries, and so what you can expect that we can do in CP. So our learning objectives are for you to understand the core differences between a clinical registry and a patient-reported registry. So we have examples of both of those this evening, and to get a sense of the benefits of each types of these registries as a resource for conducting research. And both the speakers are going to really speak to those points. And then ideally, you are able to understand and articulate what CPRN's intended level of support is for these types of registries. So there are many different types of registry. A registry is basically a database or a list of patients, patient characteristics, and potentially the medical interventions that they've experienced, and other potential data about the patients. They can have multiple purposes, so they can be just a list of people that have consented to be contacted for future research. It can be used to look at longitudinal information or long-term outcomes about patient characteristics, natural history, and what happens after many interventions. They can be used to link to biobanking for blood, tissue, genetics, or omics, these very rich data sets about people like proteomics, I'm not even going to try to say that one. Jay can say it for me. And then long-term outcomes and for quality improvement. Dr. Riva-Cambrin will talk about that quite a bit. Registry data can come from a lot of different sources. It can be either abstracted or have a person look at the medical record and put it into another database, or sometimes it can be taken directly from that. It can be done by interviewing patients, or it can be patient reported. So that's there are a number of different sources of registry data. So the purposes are, as I said, you can use it to contact people for enrollment in studies. You can use it to characterize the population for a given condition. So you can look at trends. You can understand what is the nature and distribution of the condition in the population. They can be used for generating hypotheses or like understanding good study questions to do additional studies. They can be used for looking at differences in practice across different clinical centers or different regions. And then they can be an excellent tool for determining the feasibility of a study. A lot of studies fail at the level of enrollment because they are too aggressive about how many people they can enroll, and a registry can provide very valuable information about that. So those are some of the purposes, but I think there's nothing like understanding it in the context of an existing condition. So our first speaker is Dr. J. Riva-Cambrin, who is an associate professor of neurological surgery at the University of Calgary. And I've known Dr. Riva-Cambrin for about 10 years because of our work together in the Hydrocephalus Clinical Research Network. And so I'm going to put up his first slide, and I'm going to hand Jay the mouse and keyboard, and I am going to unmute you, Dr. Riva-Cambrin, and then you'll be ready to go. Jay, you should be able to speak.
Okay, thanks, Paul. Okay, I'll try to stay within the 15 minutes. So Paul just asked me to talk about. I'm the overall PI of the registry of the Hydrocephalus Clinical Research Network, but it takes a village, and so there's also I have two co-PIs, Abco Carney and Tamara Simon, which I just want to recognize. So let me see if I can advance the slide. Not having any luck there. Let me try this way, Paul. If you could advance. Oh yeah, just a couple things for this slide, and any no conflict of interest. The Hydrocephalus Clinical Research Network really has gone over and above to try to stay clear of industry funded. research. And I should also note that the registry itself has been funded for 1 year by the NINDS and also by private philanthropy and the Hydrocephalus Association primarily, with other people tidbit intermittently funded outside of those. So just to get everyone, I know it's a CP group, but we're in close relationship. Hydrocephalus is a major cause of cerebral palsy, and the state of it in 2009. You treat hydrocephalus with a shunt, and you can see those curves there. Those are not survival of the patient, but the survival of the shunts, and those are pretty dismal curves. So 60% of the shunts are revised in 2 years. So you can imagine if you were going to buy a car, you would never buy a car that had a 60% chance of breaking in the first 2 years. So sadly, those numbers were pretty steady over 20 years. A couple major projects came out, one looking at one valve versus the others, negative. The endoscope using a camera inside the brain to see if we can help improve it, negative. And then 8 to 15% of shunts would get infected. So things are not the greatest in 2009. And so there was a white paper commissioned by the NIH, which Paul was an important, and basically, not surprising, the literature was dominated by single center studies, small studies, and if you could advance one more. And so it suggested, just like every last line in every paper is, this is a good study, but multi-center collaboration, data pooling, longitudinal would improve this, or randomized control, and that's exactly what the NIH did. So in response to that, without getting into the birth of the HCRN, and so why isn't that done most of the time? That's what the dollar sign means, is because this costs the multi-center, the data pooling, and especially the longitudinal studies is a very costly endeavor. Next slide. So the HCRN was born, and just to give you, right now we are 14 clinical centers and 1 data coordinating center, and all those dots moved in the transfer of the slide. So my geography is not that bad, but anyway. Okay, next one. Okay, so when we set up the network, the very first thing we did was come up with the registry. And so you'll see me, I shy away from the term registry because I'm a hypothesis-driven researcher, and in general, I'm not a big fan of collecting data for the sake of collecting data. I mean, that has its role. Certainly Google would disagree with me on that, and Apple, who are constantly stealing our data. But in terms of, we wanted to collect data, but we also wanted many questions in the background. So what were the principles? So I'll use the term sometimes core data project, that you could call it a euphemism for the registry, perhaps. Well, number one, we wanted to use the registry to all speak the same language. So now we all speak the same language, but when we first started, we didn't. So we'd call things different. Even simple things like this is like, it seems like nothing, but this is one of a major advantage. We merged our terminology. We now have a data dictionary, which is the heart of the registry, and common data elements. Now that's a term the NIH uses, but back when we started this, that wasn't even a term yet. But now common data elements are critical to any registry. And so we want to ensure major data fields were there for quality improvement. So buy-in is huge when you're talking about 14 disparate centers. And one of the advantages, you can always use your registry data to look to see how you're doing, and you also compare to your colleagues. And so shunt malfunction rates, and what type of malfunctions, I'll show you a bit of that. Infection complication rates as well. So the registry is constantly monitoring that, and people get monthly reports on how they're doing. Next slide. Now this is something that I would stress to anyone starting a registry. RABs, IRBs will sort of demand it, is that yes, you want to collect data, but you also want to at least have a core group of hypotheses that you're trying to generate. So when we created it, we had 4 embedded prospective cohort studies, and therefore all the necessary data fields were incorporated. And in fact, we put in several areas of duplication for data verification. currently, the registry has 9 embedded prospective cohort studies. Next one. Next slide, or next bullet. Oh, and then other fields. So we don't always know what the future will bring, so we try to incorporate any fields that we think might be important. That's always the toughest, and that's always a balance, because everyone has a million ideas, but you don't want to have the registry become too arduous, and then it'll collapse very quickly. Okay, next slide. I better speed up, Paul. So it's just slow to transition the slides here. Yeah, there's our core data project timeline. I think this is important. So our HCRN's first meeting, I believe, was in October 2006, and that was when we decided to do the registry, and I was tasked to set that up. And it took over a year and a half to design, test, various, launched March 2008, just 4 centers in the DCC, and then currently we're 14 centers, 6,400 patients, and 14,000 procedures, which in hydrocephalus is huge. In cardiology, it's peanuts, but in hydrocephalus, it's huge. Next slide. Yeah. All right, and so there's 2 major, you could just click again, Paul, and I'll do it faster, but there's 2 major hydrocephalus registries in the world, ours, and I'll compare and contrast, and the UK shunt registry. So the UK shunt registry is all patients that get shunts in the UK. So it's a population-based sample versus ours is more of a sample. We represent 2 countries, 14 centers, as I mentioned. They have 60,000 procedures. So they have a lot of quality of data, quantity, and more limited quality. Not in fact their data is bad. It's just they don't have very much information on each patient, and it's difficult to follow them. But right now, let's just read 9 embedded studies. But our registry is designed to follow each individual patient course. So we can see the patient's entire life with hydrocephalus, which is called linking, which is very important, in my opinion, for registry. Next slide. So first thing we want to do is find out if our registry is representative of the North American population, because they were high volume centers. Next slide. And so we just did a small study. We compared ourselves to FIS, which is 44 pediatric hospitals, and at the time 3 out of the 4 of us were members of them, and just to see if we were representative. This is a very early on study we did. Next slide. And you can really move the slides along, Paul. Oh yeah, and so what we want to do is compare what our patients look like versus the FIS, and race, gender, race, ethnicity, payer, all look good. They had more comorbidities, but we think that, next slide, we think that is because of coding. Those are coded immediately after surgery versus FIS codes on discharge. Next. And then similarly, we had a similar mix of between new shunts, revisions, and infection procedures. And based on this, we've published this in a response to an editorial, actually. But we've said that we think early on the HCRN was representative of the North American population, especially now with 14 centers. We definitely think, and we have small, medium, and large centers now. Next slide. Yeah, you can. Okay, so this is our accrual to date again, so 14,000. You can see each acceleration there is usually the addition of sites. And so that's where we were as of last week, actually, and 6,400 patients. Next slide. So how's the data used? Next slide. And so one thing, like especially we work very tightly with the Hydrocephalus Association. And so this is something we're interested in, they're certainly interested, like just simple things. But finally we have a definitive, like, what are the causes of hydrocephalus in North America? And there it is. So it's simple, it's elegant, and it's representative. And so we see preemie IVH, which is common to us, CP and hydrocephalus, spina bifida, and various, you can see it down there. So just a very simple descriptive statistic is crucial to planning future studies. Next slide. What another important critical thing that this good registry will allow you to do is each site has its own fingerprint. So not every site, like Canadian sites, it's much lower chance of myelomeningocele, and other sites will have higher tumor rates because of the way they generate their business, kind of like um, there'll be a national referral center for tumors, say. And so therefore, when we're doing these studies, we have to risk adjust, and we know exactly, and this case mix has stayed the same once we hit equipoise in the registry, and so now we can risk adjust per site, or we can risk adjust per etiology if needed. Yeah, go ahead, next slide. Yeah, those are just examples. We also use it for quality assurance. So identify outliers, and this is a good example. So you see, this is the reasons why shunts were failing. You could see in the disconnection group that Indian red or brown column, one site skyrocketed, and I'll unblind that site at the time I was at Salt Lake City, and that is Salt Lake City. Next slide. And so the registry twice, and I'll just give one example, has identified a major problem at a site. So this was a catheter used, and Medtronic BioGlide catheters, and this was a new thing that was supposed to be slippery and wouldn't get stuck in the brain, but it was actually, it dissolved in CSF and the catheters were disconnected. And so we saw this spike in the registry, and we also saw it at the site, and we published this as well. So that's one thing the registry can do. It can also, most importantly, probably provide pilot data. Next slide. And this is just one example. This is a third ventriculostomy and chorial plexus coagulation. It's a very new procedure that was done extensively in Africa, but we wanted to see if we could bring it to North America safely. And again, so this is a pilot, so safety and efficacy, 7 centers, just 36 patients. Just is this thing doable and is it safe? And we proved that it was safe, and we had it looked successful in North America, and it's led to 3 other major studies, of all of which have 200 or more patients in them. So another thing the registry allows us to do is that when we do this group, this is a very sexy term and appropriately sexy in medicine, precision medicine. So it's not just is this new procedure better than shunts? Well, the answer is no, but if for certain subgroups it is, and certain subgroups it's shunt. And so the registry gives us enough power to, when we do studies like this, that we can break it down by etiology of hydrocephalus and age. And now you tell me how old the patient is and why they have hydrocephalus and show me what their ventricle's like, and then I'll tell you what procedure, instead of just saying this procedure is better. That's called precision medicine, and that's the future, especially in cancer care. Next slide. But again, like I said, the heart and soul of the registry is embedded questions, so answering big prospective cohort studies that take years to move along. And I just give 2 quick examples. So the first one, you can move the slide. Tamara Simon and I looked for risk factors of shunt infection, and what it found is, you can hit the button twice, Paul, a gastrostomy tube, and then revision procedures. You can hit the button again. So if you want to prevent infection, what you should have to do is prevent revision. So then put all the money into not having to fix shunts, and infection will naturally come down. Next slide. So then the second study, which we did, and this is just showing the power of the registry. So in hydrocephalus, these were like studies of 15, maybe 60, maybe 200 out of 6. This is 1,036 prospective new patients with hydrocephalus. I had not seen power like this in studies previously. Now we could look at 33 variables, and not just hypothesis generating, but actually hypothesis answering questions. Next slide. And so what did this one say is the valve, again, the valve technology was not associated with shunt survival, so more expensive shunts do not help any better than the cheaper ones. Next one. I found a few novel things. You'll see that if you have a history of a cardiac disease, and if you advance the slide, Paul, cardiac disease, and one more time, then your shunt will malfunction sooner, and that's something that we've not really been described before, and we're zoning in on this a little bit more. And next slide. And then the use of the endoscope, that previous trial said the endoscope didn't help, but we actually proved like this shows the power of the registry doing a prospective cohort study, outpowered a randomized control study, and showed that it wasn't just not useful, it was actually harmful. And now the use of the endoscope for placing a first shunt is banned within the HCRN. Next. Okay, so that's I'm happy to answer any questions after. Sorry I rushed through a little bit, but I just wanted to cover sort of everything we've learned, everything we've used the registry for, and happy to take questions afterwards. Thanks.
Thanks, Jay. Sorry, and you were actually advancing, and so Megan, I'm going to let you try to advance with those little arrows down below, and then cue me if it's not working, but there is definitely a little delay. So our next speaker is Megan O'Boyle, and she is the principal investigator in the Phelan McDermid Data Network, which is a PCORI-funded network, and the Phelan McDermid Patient Registry. So she's going to give you some examples from a very different type of registry. So Megan, let me hand you the mouse and keyboard, and you let me know if you need help. Okay, you should have control of the mouse, and spacebar works to advance too.
Great. Well, it's nice to be here tonight, and thank you for including me in this webinar. I just want to give you a little overview because I can pretty much guarantee nobody's heard of Phelan-McDermid syndrome, and the people that named it after Dr. Katie Phelan and Dr. Heather McDermid wanted to show their respects to these geneticists, but they did not think of what the initials meant, which is shortened to PMS. So you will see references to PMS throughout the slideshow, and that is just because it's easier than saying Phelan-McDermid syndrome. It's also a way of people to remember a very rare disease because people think they know what PMS is. It was called 22q13 deletion syndrome, and it can be a deletion or a mutation of something called a SHANK3 gene on the 22nd chromosome. My daughter is up in the left-hand corner in the pink. That was many years ago. She's now 16. But it's like anything in autism and many other syndromes, there's a spectrum. There are children that can walk and talk, and there are children that never walk and never talk, and there's everything in between. There's also some varying medical conditions, including, well, here you go. The core features are developmental disabilities, intellectual disabilities, absent or delayed speech, autism spectrum disorder, and low muscle tone. Other features, seizures, decreased perception of pain, mouthing everything, lymphedema, and so on. So a lot of what we have is also falls under the umbrella of autism or intellectual disabilities, and some things like lymphedema are very specific to our syndrome. So when we thought about this, and we are still a pretty much parent volunteer-run organization, we're currently looking for a full-time executive director, but we're still in that startup phase. So a few of our parents recognized that in order to get research going, we needed to collect data, and we needed to be the group that collected the data as opposed to one doctor or researcher at one institution somewhere that may eventually lose funding or lose interest. And what I've tried to explain to our families over the years is that this data collection is not just helpful for researchers, which you'll see is the fifth bullet down, but because we return data to families, it's helpful to the caregiver, to the patient, to the medical team, the education team, of course, researchers who it was truly designed for, and eventually, we hope, industry. So as we work on improving this registry, we will need to go to both industry and researchers to say, you know, what are you interested in? But we will also be patient-centric and ask families what they're interested in. So, sorry, I'm going slow here. Paul, I still have control, correct? Okay. I think you all have already had another webinar that talked about PROs, but just for the sake of repetition, patient-reported outcomes. And in our instance, 100% of the reports are from really caregivers. But for the purpose of this we will just refer to them as patient-reported. Any report of the status of a patient's health condition that comes directly from the patient or caregiver without interpretation. In other words, a parent can answer a survey without having to tell a clinician, who then puts it into the computer. And you'll see how that works out as we go forward. So why do we have a registry? Well, there's lots and lots of reasons, but in general, we want to further research. We want to further knowledge of our syndrome, which is just working out its characteristics. And I'll explain later, but we really thought the phenotype had been figured out in the first 10 or 15 years after the gene had been discovered. And we're learning now that the phenotype is actually possibly changing in adulthood. So just learning, what does this disease look like? And finally, hoping to lead to treatments, whether they be therapies, behavior therapies, diets, medical therapies, and so on.
Hey, Megan, could you just define the word phenotype for the audience?
Oh, I'm sorry. Yeah.
No worries.
I didn't know what a phenotype was 3 years ago. If you talk about the phenotype of something, it's what are the symptoms or characteristics of a disease or syndrome or condition. So, and if somebody says, what's the phenotype and genotype, genotype would be what is the genetics involved, what genes are involved. This syndrome was first discovered by Dr. Phelan in like 1998 when she found 2 different patients in one lab that had the same genetic report. She put those families in touch with each other. A few years later, there were 20 families that got together in a hotel, and then we're at about 1,400 families in the world now. As you all know, genetic testing has gotten better and cheaper, so genetic syndromes like ours will grow exponentially just because previously children that would have been described as autistic or mentally retarded or having cerebral palsy are now being correctly described as having Phelan-McDermid syndrome, which can cause intellectual disabilities, movement development issues, and autism and epilepsy. So this is happening across many syndromes. I haven't figured out the best way to do this. So where it says PMS-IR, that's Phelan-McDermid Syndrome International Registry. Upon doing this, the only organization that really existed in the world was the U.S. organization, the Phelan-McDermid Syndrome Foundation. But we encouraged our families in Europe and other countries, other areas of the world, to start their own foundations, but work with us. But we only wanted one registry. This is an area where more is not better. More is just confusing, and more is just chopping up a syndrome into smaller pieces. So we strongly recommend to other groups that are doing registries, whenever possible, to collaborate with syndromes and diseases that are related, so you can attract more researchers and see what you have in common, or at a minimum, getting all the different groups that represent the disease to work together, even if they're groups that don't like to work together for the purpose of registries. And as I said before, we want to facilitate research, which can just be creating interest and also eventually assisting in recruitment. We want to advance knowledge about the condition. As I said, not everything was known, because all the publications in a rare disease tend to be like, 7 out of the 9 children seen by Dr. So-and-so had this. Well, neither 7 nor 9 is a very big number. So when you can take the 1,400 patients in the world and you can get 1,000 of them in one database, then you really have something that's more publishable. And the final bullet there is accelerating research to lead to better clinical decisions and better outcomes. And I think, as your webinar series topics have pointed out, those are obviously very important to your community. Sorry about this. So the bottom line is we want to make it easier for families, doctors, and scientists to learn about the condition so they can improve the lives of the people living with the condition. So how do we collect the data? So all of the registry data in the PMS-IR comes from, for now, caregivers. And we collect various information, demographics. We have questionnaires that are what we would call clinical, which is questions about seizures and GI issues and bone issues. And then we have a survey that's developmental. When did your child walk, talk, babble, and so on. And eventually, and I'll touch upon this later, we added a survey about adults and potential regression. We also have the ability to upload attachments. And in a genetic syndrome like ours, having genetic reports is vital. Some researchers won't look at data on a patient that doesn't have a genetic report because they want to know exactly what genes were involved and that it truly is the right diagnosis. In the instance of CP, you all may be more interested in imaging reports or gait lab reports. And these are harder to de-identify than surveys, but they're very valuable. So did you have a question, Paul?
No. I'm sorry if I made noise. It's slow to mute.
This is just this is our landing page. It's you can't really see, but up in the right-hand corner we have translations to about 4 different languages. Translations are tricky. They can be expensive, but the rule of thumb is you get it right in English and you test it and you make sure you have it really perfect, as perfect as you can make it before you pay to have it translated, because every change has to be made in every translation. So this is just, I know I don't expect you to see what is on the right-hand side with the arrows, but I'll just walk you through it. I said to you that there were different surveys, clinical, adult issues, and developmental. There's also, at the very bottom, you'll see a bunch of special projects. We had a researcher who wanted to send out surveys to our families, and we said, we don't let that happen anymore. We don't allow that to take place because our families were getting survey fatigue, and they never saw anything get published. So if you want to ask our families questions, you have to ask them through our registry. But the upside for the researcher is we've already recruited our families into the registry. We've already consented them to get their data and share it with other databases. And we can contact our families and say, hey, Dr. So-and-so from University of wherever is interested in our community and has posted 3 new surveys. Please go on and answer that. We would then give that researcher time to, publish on it, and then it would become our data. This is how the questionnaire, the clinical questionnaire specifically, is broken up in the survey. Our families answer questions about diagnosis, and that can range from what is your child's genetic diagnosis to who paid for the genetic testing. And so you can see here there are a lot of different areas of the body because a lot of different systems are part of this syndrome. And this makes for 100 questions in this one survey. And I was told by advisors when we set out to do this in 2010, we launched it in 2011, don't ask more than 50 questions and keep it to 25. We have 300 because we needed to understand what this disease looked like. And when I get to how lessons learned, you'll see what we've learned about doing it that way. Sorry, guys. Okay. This isn't that relevant, except that it shows you that in our registry, we can easily pull up reports that tell us where, various different aggregated information. So it's not surprising that most of our data is coming from patients in the U.S. because that's where the registry started and it's in English. But this is interesting that so much of the rest of the world has made an effort to be registered, and they all want to do translations and increase this. So that is a great sign. And in a rare disease of 1,400 known diagnosed patients, we're probably actually in the tens of thousands. It's critical to get people from around the world because the best clinical trials may be happening anywhere. It's not necessarily going to happen in Chicago or Houston or somewhere in the States. This map is just an example of after you've answered your survey questions, you can click on various things to get aggregate information. And our families can actually click on wherever they live, and it will drill down to like how many people are in their state. So how can this help researchers? Because what our previous presenter showed us was very cool. I mean, quality improvement, improving quality of life, and not having shunts replaced. That's a pretty huge outcome. So what is a PRO going to get you? And so one area is data sharing. Researchers are able to request from the foundation de-identified data, and we can also provide aggregate data or patient-level data, and I'll give you examples of that.
Megan, can you give a one-sentence definition of de-identified data?
Yeah, and I'll show you a picture of it.
Okay.
So if a doctor is interested in seizures, for instance, he would tell our registry, he would apply for data access, and he would look at the questions and say, I'm interested in male patients post-puberty that have had more than one seizure. And we could drill down and send him a spreadsheet with all the patients that qualified for his study. He would not get birth dates or names or geographic location. He would get de-identified information. Now, if he was recruiting for a certain area of the United States, then we could do something by zip code. And I'll show you an example of that in a moment. Aggregate is more, a picture of everything, less specific. We've been very successful with using the registry for recruitment. A doctor might contact us and say, I'm interested in the kids that are having ear infections and symptoms of bipolar. Well, that's really specific, and we don't want to bother, all 1,000 people that answered the question. So we can look at the people that answered yes to those two things, and then send them an email and say, Dr. So-and-so at the University of wherever is doing a study, and your child fits this. Please contact him if you're interested. So it's able to really do targeted recruitment. You can also target by genetics, which nowadays, as the doctor said, precision medicine, this is what it's all going to come to. Sorry, I never thought advancing slides was this difficult.
It's slow. You can ask me to click if you want.
That's okay.
It's a little faster if I click. So.
Okay. So this is an example of de-identified data sharing. So, it has a patient ID, when they answered it, their, age, gender, and, in this example, do they have visual impairment? This would be an example of aggregate. We have a cardiac questionnaire or cardiac questions within our clinical questionnaire. So after I filled it out, I, as a parent who's filled out the information, could look at that and see that a certain number of kids have had a heart murmur. Again, with rare disease, it's, people are just dying to get information on the community, and they don't want to wait till something comes out in PubMed, nor do they necessarily want to pay for it, nor will they necessarily understand what the PubMed article says. So returning data has been a very valuable thing for us. We try and explain to our families that they have the opportunity by participating in the registry to impact the research. The more patients that are in this, the more accurate the disease will be characterized, more complete. If they have their medical records with them at 2 o'clock in the morning when they're filling this out because they can't sleep, that's much better than if they're standing in a clinic and somebody's asking them, questions and they can't remember, and their kid is trying to get out of the door. So sometimes, PROs can be a lot more effective and accurate if you're doing them from home and you have the right records with you or a good memory. So, you know, the better the data, the better the outcome. So, limitations of the data. Not everybody has been diagnosed, so we don't have everybody. We only have what we have. Not everybody with the condition has joined the registry, but we do have 70% of our known diagnosed population, which I think Paul and others would agree is a pretty high, it's a very high engagement rate, and I would say it's because our families are dealing with a rare syndrome, and it's pediatric. It's a life syndrome, but it's identified early in life, so you know, they're desperate for information. Not everybody can answer 300 questions. They have a really good attitude, and they enroll and they fill out their demographics, and they get, you know, 20 questions in and they get interrupted, or they get 50 questions in and it gets depressing, and so they may come back and finish it, or they may not. We ask our families to come in every year and update the registry, because we're trying to make this a what's called a natural history, where you look at a disease in a patient over their lifetime. And we will need to lessen the burden for our families if we expect them to come in every year, because 300 questions was too painful, and so they put it off. It's like paying your taxes. And the final bullet is, not all registrants could find their genetic data, or they live in a country where they were never given a genetic report, they were told over the phone, and that's an issue for the researchers that want genetic data first and foremost. So I just want to give one example. We're way over time. Let's go to Q&A, and if it comes up, I'll give the example.
Okay, thanks a ton, Megan. Let me talk a little bit. We are a bit over time, so I'm going to go a little fast. And what I'll say is, in the Q&A, I'll have people ask questions of Dr. Riva-Cambrin and so Boyle, and then we will we can save some CPRN questions for sort of towards the end, since I have more time flexibility not being on the East Coast. Okay, so CPRN has a clinical registry and a patient-reported outcomes and research contact registry that we call our community registry. The CPRN clinical registry collects patient characteristics, interventions, and outcomes. So it collects information in all of those areas, and it collects the data by electronically taking it from the medical record. So the person inputting the data is the physician or the PT during a clinic visit or during a surgery. So the clinicians, if it's a surgeon and they do an orthopedic surgery, it's part of their post-operative note is done into a form that then goes directly into the registry. The sites transfer their data. Initially it was 16. We're up to actually 18 sites, but not all. We've got 18 that have committed to it, but not all 18 are implemented yet. So they transfer their data to the University of Utah into a centralized database there. And this can be used in many of the types of examples that Dr. Riva-Cambrin used in his talk, for supporting hypothesis generation, for study planning, to look at practice variation that can then be a target for quality improvement, or to be able to do that risk adjustment that he was talking about. And it uses what's called a limited use protected health information, or PHI. So Megan was referring to birth dates as an example, and so de-identified sets don't have anything that can be used to figure out who you are. We use a limited set, so there are no names, no social security numbers, no contact info in this, but we do have birth month and year, as well as visit dates and event dates if you have like a surgery. So the CPRN patient-powered registry, or PRO registry, or the community registry as we're calling it, collects patient characteristics and interventions and outcomes as well, but all from patient report. So these are entered via surveys by people in the community. It can be people with CP, or parents and caregivers of people with CP. And it is intended from the get-go, it is a long-term study. So we are setting it up with surveys that will happen once you enter the registry every year. And we're using some technology called computer adaptive testing. So it's very smart about how it asks you questions to reduce that question burden that Megan was giving an example of. And so that's critical to get that natural history of long-term outcomes. So it supports survey-based studies, and then it will also support contact information for additional research. And I should note there is a research contact database or registry already. I mentioned it in another webinar called CPRR, run out of Northwestern University. So one of those does exist for CP, but this is going to combine these two capabilities. And then the last thing that's truly unique about what we're doing is we're creating a linkage between the CPRN clinical registry and the patient-powered registry, so that researchers will have access to a richer data set that combines all this patient-reported outcomes with this clinical research. And it can be linked up without identifying patients using some fancy software to get that done. So in summary, registries are databases about people with a common condition. Clinical registries collect that data from people in a hospital setting, or patient-powered registries collect it from people in the community. Registries can be used to improve the ability to study a condition by providing this essential information about the patient characteristics, the phenotype that that Megan O'Boyle was talking about, and the interventions, what's happening medically, what's happening surgically. And then it can provide additional contact information for enrollment in studies, and then this longitudinal study, which we know is critical for a condition like CP. So CPRN has both a clinical registry and a patient-powered registry, which we call our community registry. So with that, I am going to open it to questions, and I'm going to encourage you for the next 5 minutes to ask questions of Dr. Riva-Cambrin and Ms. O'Boyle, and I'm going to turn and look at what questions we have. Okay, so Jay, I think this question is going to be for you, and I need to unmute you. The question is there a registry for microcephaly?
Not to my knowledge. So yeah, I think there are small university specific, but not to my knowledge a multi-centered group. Now with Zika, that's probably going to change quickly, but not to my knowledge.
Okay. All right. I am getting into one of my technology challenges where I can't get my questions visible. The next question is, can a parent and an individual enter information separately, or will these two, can these two entries be linked? So that's probably a question for you, Megan.
So far in our community, can you hear me okay, Paul?
Yes, I can.
In our community, we are not at a point where we have patients that are functioning at a level that could answer questions on their own. That's not to say that that won't occur. As genetic testing gets better and more specific and people use different kinds of tests, we will probably have smaller mutations and higher functioning patients. So we're prepared for that, but currently it is just one designated person per profile. I know that with other registries in like Alzheimer's, for instance, there is places for caregivers and patients separately that can be linked. I don't know if Alzheimer's links them, but I think it can be done. But for us, it's a non-issue.
Great, thank you. And I will say that that is, well, so I have questions coming in about that specifically. That is on the radar for CPRN. We know how we're going to solve it. We haven't actually technically solved it, but we haven't launched our patient-reported outcomes registry yet. It's imminent and will be launched for adults first. But you know, there's definitely the possibility to have both, and we want to be able to link those records. So we've got some design work that we need to do there. I want to encourage people, you can ask questions by either raising your hand or using the question mark icon on your control panel. I'm going to go now to a voice question from Daniel, and I'm going to see if I can unmute you, Daniel. Go ahead, fire away.
Hi, Paul. Can you hear me?
I can.
I guess I'm wondering how, I'm really quite curious, how does this relate to the patient-centered research, setting a patient-centered research agenda? I'm just trying to go over in my mind, are we thinking that the creation of another registry for CP, a more specific registry, would be beneficial to the community, or thinking of enhancing some of the other current registries that are out there? I was just wondering your thoughts are on that.
So let me come back to that in a few minutes. I want to be respectful of our speakers' time and everyone's time in general, but I want to see if there are other questions that are not about specifically CPRN. So I'll come back to that question. Thanks, Daniel. I have another question. So let's see, if there are hundreds of questions are exhaustive to complete, how do you determine the most pertinent that need to be answered?
So that's why we included 300 questions, because we wanted to see, what was on people's radar. And this somewhat gets into patient-centeredness. So we did the best we could with what we had in 2011 when we launched it, knowing that we would probably do some sort of improvement or update in the future. A little bit of throwing mud at the wall to see what stuck. And we found through a combination of the data that was being put in the registry and conversations that came out of the registry that were on social media, that there were things that we left out that we had never been written about or published about, so they were not considered part of the syndrome, but yet there was a lot of talk about it on Facebook. And so we actually added questions to the registry based on things that families were talking about. And that would be an example for us of how this became very patient-centered. The other thing is, we asked 10 questions about allergies, and we can tell that the answers, we called the CDC, and we said, what's the statistics for allergies in the general pediatric population? And our families fell within or below that. So allergies are definitely not an issue. We'll take allergies off the table. But I will tell you, when consulting with researchers, there's not many things that they were willing for us to get rid of. They wanted us to keep everything. So it's part of it is lessening the burden will be us investing in better, more expensive survey technology. In other words, right now, new families would have to answer questions about pregnancy and also about puberty. Well, if you have a 2-year-old, you should be able to not have to look at the puberty questions. And if you've already answered questions about pregnancy, you shouldn't have to look at them again. So we did what we could afford in 2011, and when we improve this registry, we'll have skip mechanisms, and that will shorten it so that you won't necessarily see all 100 questions. But getting the questions right, and as they talked about earlier about common data elements, it's really easy in theory. It's very difficult in practice, but it's important. And you just do the best that you can. But you have to at some point say, I can't let perfect get in the way of the good, and launch what you have and improve it as time goes on. And Paul, if we have time, I can stay on a few minutes. I can give an example of how something went from a conversation at a conference to how it became, something researchers were interested in as a result of the registry.
So let's come back to that if you can stay for a minute. I want to ask Jay the same question, if he can stick around a little bit longer. There is a question that I think is appropriate for him, so I'll either tee it up to him as his last, or if he can stick around a little bit, we'll just keep going.
Yeah, I can stick around a little bit.
Okay. So there's a question that I think I know for CP, but I think it's good to know how it happens in HCRN. If a patient goes to several hospitals for care for different aspects of the condition, how is the clinical information captured, or do you currently can you track a patient that is, goes to 2 of your centers? I know they're in different cities, so that may not be a big issue, but I'm wondering if you've ever dealt with that.
Yeah, no, we can only track a patient as they go through their center. Now, if the patient is saying, "Oh, I'm moving to St. Louis" or something, then we have a method of tracking. they have a different, all of our information's de-identified as well, and a different way to link the two randomly chosen IDs that we know it's the same person. Now, if they move to a center that's not in the network, they go invisible to us. But fortunately, that limitation with in pediatric hydrocephalus, patients are sort of married to their surgeon. It's a unisystem disease as differentiated by PMS, and so it's less of an issue for us. But it is always a worry that when patients can disappear, or if you have a competitive, like take Memphis, for instance, there's 4 major neurosurgical centers in that city. Then patients can sometimes move around or doctor shop or whatever you want to talk about. But in general, that's not the case.
Okay.
It's an accepted limitation.
Okay. And it's you don't view it as terribly burdensome to the end result of your data. Like, you still have a rich data set without that.
Yeah, like I mean, again, it's just the nature of pediatric illness. People like their doctor, like their hospital usually, and stick to it. I mean, we have patients that are in Hawaii and say, well, my shunts blocked, I'm coming home. And we're like, get it done in Honolulu. And they're like, nope, I'm coming home, you know. So that's unique to pediatric. If this was adult hydrocephalus, that is not necessarily the case. Patients will just often do what's convenient. Adults do what's sort of convenient.
Okay, thank you. Megan, do you want to answer your question of an example, the example that you were about to give?
Sure, I'll try and give it quickly. So in 2000, we meet every 2 years for a family conference, and in 2012, we only had a few adult patients, families there, and topic of regression came up, and some mental health regression, bipolar type stuff. And as it came up, a few families went, oh, I thought it was just my child, so I didn't say anything. And this was not something that had been characterized at all in the publications. There had been mention of it by an Italian researcher, and we thought she was off the mark because we hadn't heard it anywhere else. So, as consequently, we opened a closed Facebook group for just parents of a handful of adults, I think maybe 20 or adults. And for 6 months, those parents spoke openly on Facebook. And we wanted it closed because we didn't want to scare the other parents, and we kind of wanted to see what was going on. From the conversations on those Facebook, Dr. Phelan and one of our research moms came up with a third survey for, we call it adolescents and adults. And the questions were pulled from conversations on Facebook about loss of toileting, loss of weight, loss of sleep, things that were happening in late teens and adulthood, according to the families on Facebook. And as a result of collecting that data, we then had something to talk to researchers about. So the Facebook group came out in 2014, and in 2016, last summer, we had our family conference. And we invited researchers to sit at tables with our families for a day. It was a PCORI-funded meeting, PCORI-funded session where researchers were spread out throughout the room, and each hour was a different topic. And the feedback we got from the researchers afterwards, besides they enjoyed it, and the topics ranged from GI and epilepsy and autism to adult regression, was they came to us and said, all of our clinics stop looking at kids after they're 18. The clinics only look at kids up to the age of 18. We need to start looking at your adults. And so within a matter of months, we got some seed funding, and they're going to start a protocol for adults to look at why is this regression happening. It's only happening in certain populations or cohorts, and has there been any treatments that have been affected? And my daughter is actually 16 and has had 2 regressions in her teens. And so at the time, I was like, yeah, thank God it didn't happen to us. And lo and behold, it has. And it's a story that has unfolded that we never knew would have happened. And we were able to inform researchers about which direction they should be looking. I hope that's helpful.
Great. Thanks. So Jay, I'm going to answer a question here, and then I want you to just augment it if I'm missing anything. Someone asked to define omics data. So there's the proteome, there's the genome, there's the metabolome. And so the idea is, like, for the proteome, genes produce mRNA, which produce proteins. And so the proteome is characterizing the proteins that are found in a tissue or in a fluid, and that can be used as a marker, just like a specific gene can be used. So there are all these large databases of things that are called omics because it's the proteomics or the genomics. Jay, I know we're doing this with Dave Limbrick's work. Is there anything you'd add to what that is and why it's important?
Yeah, and then metabolomics is those proteins get broken down into other compounds, and sometimes you can't measure those proteins, but you can measure those metabolites of the protein. So it's all just which stream you want to check. Yeah, no, we're getting more and more interested. I mean, we are a clinical research network, but you just sort of naturally drift. We're always looking for biomarkers, which can be any biomarkers. Don't have to be genes or proteins. They can be an MRI. They can be a simple blood test. Anything that can predict how someone's going to do, in our case, intellectually and functionally throughout life. And we are in the proteomics world. We're not in the genomics world. And so we are now introducing that a CSF biobank and linking it to eventually linking it to the registry. It's going through an evolutionary process, not a revolutionary process at the moment. But
great.
So
Paul, go ahead. What were you going to say?
I only talked about our PMS IR registry. We do have a database that our registry feeds into at Harvard that's funded by Bakori, and it takes all of the information that we collect in that registry and it maps it to 20 different omics. So, you know, the question may not have been asked about metabolomics, but there's a question that relates to it, and the epidemiologists at Harvard can, you know, map these things to all these other things. So when researchers look at the data from that angle, they're getting it in kind of the language that they understand, not in the way the question was presented to the families. And that database also uses medical records that families have uploaded, but that's a whole nother webinar.
Yeah, and this is sort of the big data slice of all of this disease research. So I'm going to pause and say thank you very much to Jay Riva-Cambrin and Megan O'Boyle for their presentations. There are a number of questions that are about the CPRN registry capabilities that I want to answer. I want to free you guys to go back to your families, and just really appreciate all your work going into this and taking the time tonight to present this to everyone. So thank you guys both very much.
It's
Thank
my
you.
pleasure.
Best of
Thank
luck.
you for the opportunity. Yeah.
Okay, thanks. So I'm going to try to go through a number of the questions here. So is the registration process open to participate in the registry for CP? So I don't know if that's a question about the CPRR, but that one I do believe is open. The CPRN registry, there's of the two. The clinical registry, you just get put in as a function of going and visiting a clinic. We're up and running it nationwide today. We've got institutional review board approval at 11 other sites. What we're doing will happen slowly at some, but then it'll capture a lot of data because it's going into the electronic medical record. And then we have some where it's being done in the way that HCRN does it, which is with chart abstraction, and that we've got about 4 that are about to come online because we just got our IRB approval for those. So those, you don't have to do anything, that just happens. And for the community registry, when we launch it, it will be open for people to participate. And I will also say to the question that Jerry Cameron answered about patients moving between centers, we believe for CPRN, because we're going to have this global unique identifier that doesn't have any patient information, but it's associated with it, but it allows us to track patients between centers. we should be able to get information if they receive services at different centers if they're part of CPRN. And our goal is to be very, very broad because of the way we're implemented. There's a question from Amanda. Would these registries provide families and patients with completed questionnaires for them to share with healthcare providers to avoid filling out multiple questionnaires with multiple providers? So there's two sides to our patient-reported outcomes mechanism. The first one that we're doing is the community registry, and yes, you would be able to provide those. So you'll be able to print out your report and take it to your medical visit. But we're actually also working the other side of it, where it would appear in my chart or whatever the mechanism is with your provider, where they can allow you to take surveys maybe before you come in or do them online while you're there, maybe on your smartphone, where you fill them out once and then the providers can see the information right there. So we're working on both of those, but it's just quite a bit easier to do the community registry first. Moving along, if you're coming into the registry as an adult, how hard is it to link that to documented pediatric information? So we have not tried to solve that problem yet. There's a real dearth of adult research, and Mary Gennady and Debbie Thorpe, who lead our adult registry, felt like there was plenty of work to be done just working off of a little bit of what people remember, but mostly what is their current situation, what is their current level of pain, what, some social and emotional questions, employment questions. So we're starting there with adults. We're not working on linking. Ultimately, there's lots of technology that can be applied to scanning older records and whatnot, but I'm not clear that we're going to make an investment in that any time soon. But there are technologies to help with that. Okay, moving down. Jay Riva-Cambrin, are you still there? Because if you are, there is a question I should have handed to you. I don't see, let me see who I see. I don't see you. He took the time to be with his family. So the question was asked, what is practice variation? And so practice variation is, you might go to a center that that focuses on baclofen pumps for dealing with spasticity, and another center might focus on selective dorsal rhizotomy, or some will be very focused on orthopedic surgeries, or some are very focused on physical therapy and doing it in different ways. So those are all differences in practice, and those probably yield different outcomes. And so being able to track the difference in practice across centers can be very useful for finding things to standardize, for doing that risk adjustment that that Dr. Riva-Cambrin was talking about, because you might have, he referred to the marketing, the way your business is marketed, because in neurosurgery, which is a big revenue generator for a hospital, they may be doing more tumor patients, and that may generate more of a certain type of patient. And so being able to look at practice variations is a very critical thing. Okay, let's see. Are there any I've got a question that's an audio question that I'm going to take. Amber, I see you there. I'm going to try to unmute you. You're self-muted. So Amber, if you want to try to unmute, I'll let you ask your question. And if you're off because I didn't notice you there for a while, I apologize. No, you're now unmuted. Amber, do you have a question?
I did, Paul, but it's okay. It was I wasn't sure who it was for. I was just wanting to clarify that my understanding is that the registries will enhance precision medicine to increase individual care or quality of care, so that patients and clinicians and researchers will be able to give information and get information. Is that correct?
Absolutely. And when we talk about it, like if you look at what was popular on CoDigital and whatnot, I mean, I think there's a clear, everybody kind of wants the algorithm or the formula to say, given these patient characteristics, which interventions at which times yield the best outcomes. And that's about getting much more precise, as opposed to, everybody that gets this and then gets this. It's like figuring out the precise way based on the input, and some of that could be based on genetic information, but mostly it's going to be based on other patient characteristics, and maybe genetics will get added over time. So that's exactly correct.
Okay, just want to make sure I understood that correctly.
Yep. Okay, so Daniel, another question, which is, are there any privacy issues or negative consequences of using Facebook for discussing health information in a private group? So I'm a strong believer that that is somewhat risky, although it certainly happens all the time. You know, if you don't have your privacy settings set right, people can see what groups you belong to. So if you feel like you might be discriminated against, if you've got you know, a medical condition and people can see that you subscribe to groups that are associated with that medical condition, you could be discriminated against. And so I think there can be issues, and that's why when we look at doing things like our registry, we do a portal with sort of obfuscation, and there's no way to get at, like, the privacy of all that is absolutely critical to what we're designing. So with that, I think we've gotten through most of the questions, and we are now at 6:18 here on the West Coast. So I'm going to just say thank you to everyone for staying this late, listening. I hope you got a lot out of this one. We'll be getting back to you soon, but please take your survey, and if you have any unfinished webinars or webinar surveys, please take those as soon as possible. And we hope to send you positive mail on Monday, April 10th. Thanks, everyone, and thanks again to those our speakers. I think they did a great job giving great information about what we can do with registries. Thanks, everyone, and good evening.